All right, cool. Hey, so it's 12:30, so I would like to start. Thank you everybody for joining in person and on Zoom. It's lovely to see you all. So it is my great pleasure to introduce Dr. Rob Guralnick. He is the Curator of Biodiversity Informatics at the Florida Museum of Natural History in Gainesville. And Rob does pretty much everything under the sun. He's got projects in modeling, landscape genetics, molecular phylogenetics, phenology, which you're going to hear about today, data accessibility, making databases, pretty much everything that's cool, that I think is cool anyway. So that's why I invited Rob to talk to us today. And so yeah, take it away, Rob. Thank you.
Hey, thanks a bunch for having me here. It's actually great to be in person. I can't believe it's been this long since I've seen a group of people in the audience. I actually went to my first in-person presentation as a master's student defending in Florida last week. It's wonderful to actually get back to that. And I will admit though that getting back to that also has its challenges, like remembering things like how to use a pointer, like how to advance slides without it happening on Zoom, all those things that I have forgotten about. I'm hoping to kind of relearn here, and I hope for your patience as I relearn it. But what I wanted to do today is cover a bunch of work that I've been pursuing in the area of phenology. And when I was putting this talk together, and Kristen asked me for a title, what I really wanted to do was frame this in this context of times arrow, times cycle, and time scales. And that's represented up at the top of the presentation here.
You should share your screen.
Oh, I'm not doing that?
Not yet.
Okay. Do you want to come up here and I got it. I can do this. I remember exactly how to do this. Just here we go. Wait, maybe I don't know how to do this.
And then you're gonna have to pop it back open. So there's like a green.
Go for it.
Okay.
We'll get this off. Sorry, guys. See, this is what happens when you go hybrid, right? You learn all these important lessons of what you should do before you start your talk. But bear with us as we get this started.
All right, here we go.
Okay, great. Can everyone see this? Can everyone see the slides on Zoom? I'm looking for, I guess there's no one visible, but nods or pluses on the chat. I think we're good. Yep. Thumbs up. Great. All right. Awesome. Okay. So back to where I was at the start of this. I kind of framed this talk in the idea of times arrow, times cycle, and time scale. It's represented up here. And the idea of mentioning this is that my presentation is going to cover a lot about phenology. And I think phenology beautifully encapsulates this idea of times arrow, times cycle, and time scales, because when we think about phenology, we're often thinking about things like phenology trends and how phenology is changing, year by year, or over longer time scales, right? It could be we're talking about phenology over millennia, and then we're kind of extending those scales out over time and looking at these trends. But phenology also nicely illustrates the idea of time cycle, because phenology is really the cyclical events that happen over seasons or over years. We think about this right now, looking outside this window, and we're thinking about phenology all around us, right? We're watching the leaves turn yellow, senescence trees. These are annual events that happen cyclically, right? It's a time cycle. And I also love focusing on phenology as a presentation, because I think, when we think about the importance of phenology, it really is one of the three organismal responses to global environmental change, right? And so we think about distribution changes that are happening in the face of disturbance and change out in the environment. We think about phenotypic change that happens to species, populations in the face of environmental change. And phenology is really the most proximal of all of those changes. The real fingerprint of change is felt first phenologically. That's where we're seeing these changes happen most. And it's super important because those changes are happening differentially across trophic levels. And so what's happening to the plants in terms of their sensitivity to climate might be different than what's happening to the insects that require those plants as a food resource, as a consumer. And if those phenologies are not aligned, they've been evolved to be aligned, and if change is slow enough, then they're going to be aligned because basically the available food resources will drive that synchrony of those phenological events between trophic levels, right? But over cascading change, we might actually see those disruptions in the timing of these events and the phenological asynchronies and mismatches that are really important for organismal response to change and basically demographic and fitness consequences. And so in putting this talk together, what I wanted to do is start with a vignette that covers this idea of phenological asynchrony or phenological mismatch. I'll use both terms, they're really different to me. Asynchrony is just a pattern. The mismatch is when that asynchrony has a consequence in terms of fitness or demography, right? And so we'll cover a little bit about this in a tritrophic kind of framework where we'll talk about vegetation phenology, insect phenology, and bird arrival patterns and migratory movements. And then for the vignette at the end, I'm going to talk more about using large-scale phenological data that are built through infrastructural advancements to kind of assemble phenological data to ask questions that are more about species and their responses to change. And we're going to really focus on how urbanization and climate together impact phenology at wide cross-continental scales. And these are bookend vignettes. And in the middle of this talk, what I really want to do is cover infrastructure and talk about building the tools that allow data to be assembled, interoperated, and synthesized, and how hard that work is, how important it is, how needed it is. But I want to really bring it home to the idea that that work isn't just about building infrastructure for infrastructure's sake, but building communities and building capacity to enable open data and open science. And to me, that is a critically important part of what I do. It's not simply infrastructure because it's fun to build tools. It's like we need to basically capacitate people broadly across a wide spectrum to be able to access those resources and use them. And that's no small challenge at all, as many of you in this room actually know. That's like one kind of meta story in the presentation. The other meta story that is sort of latent across what I will talk about is the power and importance of community science in the work that I do. And the importance of community science, not just as a way to kind of assemble larger and larger data, but to be involved in the community science aspect the whole way through. I use the term citizen science up here, and I'll if anyone's interested over a beer or coffee or something, I'll be happy to tell you why I use community science more and more and sort of stay away from using citizen science all the time. There are lots of reasons for the word choice. And finally, as all of you hopefully know, both on Zoom here, hello, and here in the room, I care really deeply about digitized and non-digitized samples, the actual specimens that we manage in collections, and they're vast importance for understanding our legacy of biodiversity that basically drives a ton of my research. And yeah, digitized collections data are super important for what I do, but if you want to talk about the work I do running an ancient DNA lab at UF, at Florida Museum, or you want to talk about large-scale work we've done to kind of build genomic resources from museum specimens and doing that in a constructive and as non-destructive as possible way, I'd love to talk about it. But I really, really want to bring home the importance of samples and the digitized data around those samples for the work that I care about. Okay, with that all said, let's jump in here. If I can change slides. How do I do that? Should be good, right? So just clicking over here. Yeah, okay, I got it. We're good. It's so funny being in person. I remembering all these things about clicking buttons I forgot about. Okay, so what I'm going to cover here is phenological mismatch across trophic levels and focusing on North America. I mentioned this, already, so I won't dwell on this, but basically, the phenology really is the study of the periodicity and timing of biological events and how it relates to climatic change, both sort of seasonally and interannually. And like I had mentioned before, I think this periodicity and the cyclicity is really worth thinking about in terms of its importance in terms of these biotic interactions. But also, phenology is just really important as it has cascading impacts in terms of biogeochemical cycles and ecosystem services. So when we watch leaves fall off the trees in fall and we think about the nitrogen that is being put back into the system, or we think about treated leaves appearing and unfolding on plants in spring and the carbon that's produced. These are really important. These are really important for how ecosystems function. And I want to mention here that the work I'm going to present is work that was originally led by Stephen Mayer, who was a postdoc in my group. This came out of a workshop that happened about 2014. That was the ESA organized workshop for early career scientists about scaling up. And the ideas about looking at phenological mismatch across scales really came out of that workshop. I was involved in helping to organize it. And watching this great group of early career scientists sort of coalesce to sort of think about the challenges of really bridging scales, temporal scales and spatial scales, and looking at phenology was awesome. And what's even more awesome is that led to this long journey, 7 years later, where we're still getting these kind of big resources together to answer questions about phenological mismatch across scales. Super fun. So regarding this phenological mismatch hypothesis, this is a really sort of important through line of this presentation is thinking about phenological mismatch. So we're really trying to figure out if climate change is disrupting phenologies differentially across different levels. Different species and different clades have different sensitivities to climate forcing. And so what we're trying to figure out is, can we look at those overlaps or lack thereof, look at how they're changing the trends, right? The times arrow here. And then if that is what we're finding, does it have negative fitness consequences? And can we test this across broad scales? So I'm going to focus here on two parts of this, the story, the vegetation part, the spring green up part for plants, and focusing on birds and their migratory arrival times into their summer breeding grounds from their wintering grounds. And importantly, I want to mention that arrival is not the peak when birds need the resources. They need those resources when they're basically breeding, right? So that breeding period and that period where they're feeding young are really where they need those peak resources from the insects. They need to align their arrival to make sure that peak availability of insects are there for them to feed their young, or else there are certainly going to be consequences for the birds, right? Okay, regarding the insects, this is where I've actually focused my last 4 years of effort on these projects. And insects are super fascinating. I'll touch on them really briefly at the very end here, because I think this is one of the key questions about insect declines and the drivers of insect declines globally. It's a totally fascinating topic. Let's start with the vegetation phenology. So here's just a look at average green timing across North America. This is for 2001 to 2012. This is based on remote sensing data that gives us a measure of basically onset of photosynthetic activity. And what you expect to see here is like this green wave, right? As you go from south to north, you should see earlier green up in the south, and then the wave of green up going north. And that's exactly what you see. So we're looking at Julian Day here, and with dark green being earlier spring onset, and yellow being later. And you can see this sort of green wave. It's not purely latitudinal. There's lots of longitudinal variation, a lot of topography in North America. But importantly, what we really want to do is look at the trend, right? We want to look at the time zero of green up. And here you can see this sort of almost pointillist Seurat painting of North America, where basically in the eastern temperate forest up into the boreal, you basically have a signal where spring is getting earlier in terms of the beginning of green up, and that's shown with our red pixels. And in areas in the west, actually, we see sort of surprisingly a signal of potentially later green up, right? Which is not what we maybe expect to see, but this is a very different kind of system in the west. There's real differences in water limitations and other reasons why we might see a signal like this. But for at least the eastern US and boreal regions, spring is getting earlier as climate's forcing spring to happen faster, right? So, right, so we have the data we can use to look at the trends in vegetation phenology, right? That's great. How do we get a handle on when birds arrive into the breeding grounds? And this is where community science data are really powerful. So how many of you have heard of Ebird? If you haven't heard of Ebird, it's great. It's a community science platform in Cornell ornithology labs. And what people do with Ebird is they'll go out and they'll make a checklist, right? They'll go to a spot and they'll say, yeah, I'm going to checklist a set of birds and they'll list all the birds I saw and how long I looked and where I was. And if that checklist is complete, you can infer non-detections. You can infer which birds weren't seen, right? If that's a complete checklist. So you can basically aggregate those data into grid cells across the continent. You can arrange them by time, and you can basically look at a logistic regression. It's a little more complicated than I'm showing here, but it's close enough for here. And if you want more about sort of the vagaries of trying to fit complex logistic regression values and how these tail off and how to kind of fit the tail of this as well, I'm happy to go into more detail. But basically, it gives you an inflection point for the arrival, peak arrival right here, right? So this is for one grid cell for one species in 1 year in North America for the house wren, but we can do this for all the grid cells where we have enough data, right? So from that simple idea, we can basically calculate arrival for a species per year, per cell, we can look at the green up by aggregating the green up data from the vegetation phenology, and we can actually get a measure of asynchrony, right? Simply green up minus arrival minus green up, and you have an asynchrony measure with red being higher asynchrony and the yellow colors being lower asynchrony, right? You can do that over many years. So here we're looking at the same house wren over 12 years of data, more in the present, or I guess 2012 doesn't feel like the present at all anymore, does it? But more in 2012, less than 2001, which purely represents the growth of Ebird over time as more and more people have checklisted and more people are involved in sharing their data, right? Okay, so what do you do with these data? Well, what you can do is you can basically plot arrival date in green up, measure asynchrony per species, and here's how I think about this. So like, if spring is getting earlier, that's so the slope is decreasing, right? So you're having earlier springs over later years. So your plot looks like this, and birds are basically arriving at the same time they were over that whole period. They're not tracking and sensitive to that green up. That's increasing positive asynchrony, right? So the other possibility is that spring is getting later over time, as opposed to earlier. Birds are still not tracking that green up, not tracking it, and that's increasing negative asynchrony. So there's two kinds of asynchrony. They both show up in here, and this is just showing you that idea. Hopefully you can sort of see that you have increasing positive or negative asynchronies, and you basically have the same sort of line in terms of the asynchrony pattern, but it depends on the nature of whether spring is getting earlier or later, right? Does it make sense? Easy? So we looked at this for 48 species. These are all insectivorous birds in North America. They're all commonly observed. They're all birds that have good trait data, so we can sort of learn more about what these species do and how they work. And we basically plotted, what is the pattern over all these species in North America? And what we find is that, basically there are a lot of species that have increasing positive asynchronies over time, shown here. There are some birds that have increasing negative asynchronies over time. So here are the arrival trends and green up trends over on the top here. This is the sort of summary of all these species, right? With some that are significantly increasingly negatively asynchronic, some that are increasingly positive. And so, no surprise, that totally drives with what we know about that pattern I showed you at the very beginning here with vegetation phenology, where in the West, we see lots of indications of increasing negative asynchrony for the most part, not entirely, but mostly. And then all of the positive asynchronies are in the East, where we're seeing earlier spring greenouts, right? So that's purely a pattern that's related to spatial results. And if you kind of aggregate that into ecoregions and plot that out in B, you can see that there's a significant trend in eastern temperate forests for basically this positive asynchrony, and a weak but road trend in western forests where this negative asynchrony, right? So that's super fun, but what is missing here is really getting a handle on why. Why are some species showing these asynchronies? Why are some species not? What are the traits that define how that works? What is making up that pattern out? And why are some species not faring poorly, and some species are faring actually relatively poorly in terms of timing their arrivals, right? So we didn't know that, and we didn't know the demographic consequences. It doesn't matter, right? This is purely looking at asynchrony. So we published this paper in 2017 with Stephen Mayer as a lead author, and we then were lucky enough to get a macrosystems grant that kind of took this forward. So we had shown that this was something you could look at large scale, but what we didn't have are all those important details about the demographic consequences, and we didn't have anything about the insects, right? The middle layer, the sandwich meat between the bread of the plants and the birds, right? And we didn't have the breeding dates. We only knew the arrival times. So we wanted to put that all together. What's really cool about this is it's a great group, super exciting. Actually led out of the lab of Morgan Tingley now at UCLA, but a whole bunch of collaborators involved in this project that are really great. And really the idea is to look at these local scale mismatches, see how those emerge into these macro scale feedbacks at the regional level, and then also think about teleconnectivity, where we're looking at wintering grounds for birds, summering grounds way up in the north, and how teleconnectivity might matter for what happens here. And across all this is temporal scale, thinking time, right? Which is what I think about every single day as a biologist. It's happening temporally. All right. So one of the really nice first outcomes of this work, a couple of years later, is a paper in Nature Ecology and Evolution by Casey Youngflesh. He's a postdoc at Morgan, and many of us were involved in this work. Well, we basically extended this framework that we developed in that first paper to really get a measure of sensitivity. And sensitivity is that magnitude of change in bird arrival per day. But you can just think about that as birds that are sensitive are tracking green up. Birds that are not sensitive aren't able to track the green up effectively, right? That's the key measure of sensitivity. And for these four kind of illustrative species like tree swallows and various vireos, we can see that these species are generally tracking green up, and they're certainly tracking it better at the northern latitudes than they are at the southern latitudes, right? There's more reasons to want to track those green up phenologies in southern latitude, tracking the green phenology in the northern latitudes, so the shorter growing more important, right? And we can see that like northern parula and prairie warblers are not so good at tracking the green up signal. So these have less sensitivity and they actually have negative slopes instead of positive slopes over latitude. So lots of variation here. But what we're able to do is to aggregate these data and then look at traits and explain these patterns. What's really exciting is that there's one set of coordinated traits that basically picks up the signal of this differential in sensitivity. And it's no surprise to me. So we're looking at here are all 56 of these species across Eastern North America. We're looking at sensitivity at the species level. And you can see the sort of negative slope here for the pattern. And this negative slope is related to this trait score on the x-axis. And what this is really representing is that birds that are migrating faster, are arriving later, and overwinter further south are less sensitive than birds that migrate slower, are arriving earlier, and overwinter more in the north. So neotropical migrants are making these long distance migrations, coming in faster, they're coming in later, and they're not able to track green up nearly as well. They're also the ones where we see the most evidence of declines in their populations in terms of their demographics. So this is a nice story, and by the way, just to be clear about this, this was originally done without a phylogenetic correction to sort of account for phylogeny. But when you account for phylogeny in this pattern, you find the exact same results. So you find they're very, very modest differences, but the pattern is still completely consistent with what we're showing here. So phylogenetic correcting is obviously really important for these comparative kinds of studies, right? Okay, so that's exciting. And I think what's really, really awesome and really making me happy is that we're also finally able to get to the insects. And here we're actually pulling data on surveys of butterflies, adult butterflies across Eastern North America, and the same grid cells that we use for the study for looking at bird arrival. We're able to actually get a signal of phenology for butterfly onset for adults across scale. And with those data, we can begin to link up phenology abundance relationships in the insects, relate them to the plant phenology, and tie them to the birds. We're finally able to make that full linkage across these tritrophic levels. And it's really, really excellent. We're just now getting these results together and out there. They're going to be, I think, really important because the insects are the hard part, not the birds and the plants. It's really, it's been a labor of love to get this together and work this through. But hopefully next time I'm out here, I'll give you the full skinny on the exciting results here. All right, so I'm going to shift gears and talk about something that bugged me about the work we did with the birds in the plant. So bugged me was we didn't have species and we didn't have phenology of the species. We're looking at phenology from remote sensing, right? These are basically pixel aggregations that tell you about green measured from space at a spot. So we don't have species in there. We have basically a sea of green, and we're not quite able to get to that level where we're seeing individual trees in a canopy using these tools. What we really want to do is pull together all of this amazing observation data on phenology, the plant phenology that's available. There's tons of it out there, but here's a problem. When you go to aggregate those data and assemble them into a coherent whole, you can't. There are so many difficulties with interoperability that it's literally crippling for anybody to sit down and figure out how did someone over here score these Byzantine scorings of plant phenology and then do it over here. It's really, really hard. And this is a real problem. What happens is that basically people have their own methods, build their own systems, and then publish them in siloed databases, right? And they might be available or they might not. They might have licensing restrictions, they might not. So people make these things hard to get access to and often put barriers in the way of basically using the data most effectively in the community. So what you want to do is you basically want to build a reporting system that allows you to kind of standardize these data, map them to what I'm going to describe more as ontologies. And these ontologies are really handy for interoperability. In fact, they're critical for interoperability. And what's super cool about ontologies is that they're basically a form of symbolic AI, and you can infer new information about the data that you didn't have without the ontology and semantic layer that helps you basically do inferencing. And so these are really powerful tools when used properly. And then what you want to do is you want to make that available openly, and you want to build graph databases, and you want to put on make available ways to access those databases via APIs and then wrap them up for people to do data science-based workflows, right? So this is what we've been doing for a long time now, trying to develop these kind of coordinated resources. And it starts for me actually in 2016 in Fort Collins in the middle of winter. This was a group of people who got together to sort of talk over how to build interoperable phenology data. We were super lucky to get to do that. It was a chance for me to go home after my first year in Florida and drink a lot of really good beer in Fort Collins. In fact, I brought my Odell's sweatshirt along for the trip here to hopefully drink some beer with y'all. And I just did pull a y'all, so that's an indication of how long I've been in Florida. So the plant phenology ontology, I'm not going to bore you with the details on how to build an ontology. It's actually a relatively cumbersome process, but the ontology idea is really super straightforward. You basically define a set of terms, definitions, and logical axioms that can be used to describe the phenology traits of plants. You put that online in the way where there are identifiers or URI, so you can find those definitions in the logic. And you can, what you really want to do here is leverage the power of being able to use the hierarchical descriptions of these phenology traits in a smart way. And so, for example, when I say there are 10 flowers on a plant, you also that flowers must be present on a plant, right? That's a simple deduction, like the simplest possible deduction. But if one system is reporting a number of characteristics on something, and another system purely is reporting presence, having the ability to have a system that basically walks those hierarchies to find and show data across those hierarchies really easily, so you can search anywhere there and get the content at all the levels, is incredibly valuable. So to give you an example of the plant phenology ontology and kind of how it functions and works, it's actually really super simple. There's a phenology observing process that has a specified input of a whole plant. It has a specified output of a measurement datum that has a lower or upper count. This measurement datum is a quality measurement of basically unfolded true leaf presence. And an unfolded true leaf presence is a plant structure presence and a plant phenological trait. So that's a very simple description of the kind of the logic and semantics of how we view the PPO. But once you know that there's an unfolded true leaf presence that has an upper or lower count of 10 and 20, you can also infer that unfolded true leaf must have been present as well, right? So this this basis for building an ontology let us build a system where we can integrate all of these different data resources that had so far deeply resisted any integration. So we're able to bring in data from across a huge set of networks, and we are in process to do a lot more of this work, so that we can develop basically phenology observations that are interoperable out of the box across continents, which is cool. And you can basically search on this. You can go to plantphenology.org. You can basically find your heart's desire of phenology observations for any species and any phenology traits, and basically get that data easy peasy. You download it all you want. Access it through our PPO package. You can get it on through the R bindings, and you can also map it up, right? And the underlying data store is semantic, which means these data are getting, users are getting data that's built interoperable. And they never have to know that. They don't need to know the sophistication under the hood. They just need to know that when they search for open flowers, they're getting all the open flowers we have data for, no matter how it was reported originally. Very cool. For me, it may not be cool for anybody else. And we're going to grow it, right? So we're growing this integration system. We thought about building not just these ground-based observation data, but what about herbarium specimens or data from iNaturalist? How do we build those resources? How do we leverage machine learning approaches to basically help us annotate these kinds of reports? And then how does that all feed into a data integration pipeline and build it out? So I want to show you a couple of examples of that. So I mentioned this to a couple of you over the course of the morning. I run a community science platform. We're built on top of the Zooniverse. How many people know the Zooniverse by chance? Anyone heard of that one before? Yeah, it's awesome. It's awesome. Okay. Yeah, it's a cool thing, right? So like basically the Zooniverse is this platform for people working mostly with digitized data, right? And so it started with astronomers. They basically had all this astronomy data. They could not possibly annotate all the objects they're finding in images. They ask people to help them out. They're both a platform where anyone can come in and kind of participate, right? Take part, be able to help in the science. I've been running this platform for 7 years. It's a labor of love. I talk to volunteers all the time. They have millions of questions about how to do things, like how do you transcribe museum records? How do you annotate flowers on specimens? It's super fun. We've actually just crossed 4 million transcriptions last week. So that's a big milestone for us. It's a lot of work that's been accomplished by volunteers. And often those transcriptions are like the whole label of a specimen. It's not like just clicking a button. It's like I wrote every single thing I saw on this label. Longer story, we actually have the funding to basically pursue a lot more like machine learning and automation with humans like doing the work where the humans are where human effort is the most reasonable, where like labels are hard, but where labels are easy, like let computers take care of the easy stuff, let humans do the stuff that requires a brain power. So we have had multiple phenology expeditions that have run on Notes from Nature over the years. They're very simple. You come in, you see an image of a herbarium specimen. You know, does this thing have mostly open flowers or no flowers? You basically click a button, and you can annotate these records and move on. And we were really interested, like, how well do people do at this task? Like, how are we asking people to just do something that they can't do? Is the data usable? Are we able to actually open this box up to have humans come in and help us annotate these phenology reports in a smart way? Or should we just like stick with like trained volunteers in our labs? So we ran a study. This was by Laura Bernskeli. She's actually defending next week. I'm super excited. She's my first PhD to defend at UF. And what she was looking at here was basically how well do people do these phenology tasks. So here we're looking at in-person volunteers. These are people who are trained in person. These are actually botanists, and like no offense to former nearby botanists who were around this area for certainly the time. Those botanists did the worst of anybody on this task. Is this talk being recorded? Anyway, in-person volunteers actually did really, really good, right? They did really well. So they got like 97.5% accuracy. That's excellent, right? 97% accuracy is really good in this task. Can you find flowers? Can you find fruits? Can you find leaves on these specimens? Yes or no? And it turns out with herbarium specimens, where like flowers can be hidden, they can be obscured, they can be like a little like flower down in the corner that no one's looking at. Getting 97.5% accuracy is great. But look, like online volunteers didn't do that bad, right? They're at 95%. So if you give someone training materials to do work online and tell them how to do it and stick with them and helping them through it, they do pretty good, right? 95%. And if you triple score those, so instead of having just one person do those annotations, you ask 3 people to do it, take a majority rule, they do great, right? They do like basically just as good as an in-person volunteer. So this is cool because it means that like we can scale this up, we can ask people to help with this task, and get great data from that effort. So we thought about this, like, so cool, we can get herbarium annotations. We'll just push them into our system. We have an ontology system already built, and it works great. We'll just squeeze those through, and so we'll have all this herbarium data living with all this observation data. This is a beautiful world, right? We have more data now coming in from multiple sources where there's one spot to go to find it all, right? Which is great. And there's not like one spot, because basically you can have it in 100 spots, but it's semantic, so like it all fits together in this sort of semantic web framework. But there's one bug of it, and I just want to mention it because it's cool when you think about like the importance of being careful. Be careful here. What we realized is that if you have a herbarium sheet of one of our trees out here, we're looking at the maple, and you have a herbarium sheet of that, and there's not a flower, does that mean there wasn't a flower on that maple? Does it mean that? You don't know, because you have one portion of that plant. You don't have the whole plant. Look at the flower that was hiding, right? So you want to basically recognize that like absence is hard here, and presence is something that you can definitively measure. but you may not be able to measure the upper bounds. So here's that same model. And all we did for herbarium data was we added basically a simple addition, a portion of a plant that is or was part of a whole plant. And because we have that statement in our ontology, when we get a measurement datum that is between 10 and 20, we can know the lower count. We know there are at least 10 flowers. We don't know the upper count. And so we can report that with fidelity in our system so people aren't misled by the data we have. Okay, so that was important. And that's also work that's done by Laura to kind of reconfigure this. And when we do that, we can do some cool things. Like, and this is not the analysis I want to present to you because we've thought harder and harder about these data. But as just a starting point, what this is basically integrated observation data from observation records from the National Phenology Network with herbaria data that stretch much farther back. And although I wouldn't put much stock in this line because of observing effort biases towards the present, that is probably the main reason for the decrease here. But we can at least begin to integrate those data. We can integrate together herbarium data, observation data in one framework, which is really cool. And we did some stats, but ignore those basically. All right, one last quick thing on the machine learning. This is something I really love on the plant phenology systems. So machine learning approaches. So basically, once you train, basically have enough training data from annotations that come from citizen scientists, community scientists, can we actually just forgo doing more of that effort and let computers take it over and do it well, right? Can computers basically learn to annotate flowers on specimens, specimens? So and a big question for us is like, how many specimens do you need? And if we can do it successfully, that's a huge time savings for computers to do that instead of having humans do it, right? So we built basically a very traditional in one sense and very much not traditional in another sense convolutional neural network approach. These have two steps. There's a feature extraction step. You're taking images and basically kind of filtering them and taking pieces and parts and features to kind of squeeze out information about these features in the image in an effective way. And then a classification part where you're really trying to see if you can then build a iterative, basically regression-based model to do a loss optimization and get the best possible kind of result as in relation to your real world data. And so you're basically using these width height density data of your images. You can feed this, basically a set of images. In this case, we're using IDBio data for Acer and for Prunus. And we were interested to see how well we could perform. What we did here is a little bit different than traditional CNN. We also did sort of tiling and then used the Bayesian framework to basically redistribute the tiles in a smart way based on where flowers are in the plants. But long story short, we built a system that we thought could be performant for those two genera. And just a punchline before I give you some other punchlines, this has led to some recent funding to try to scale this up and do this broadly for all flowering plants. And so we're now trying to see if we can build one system that can basically properly annotate any flowering plant picture, both herbarium and a naturalist, because the herbarium data is awesome and important, but there's gazillions of images being taken by very committed community scientists, not naturalists. So I have a question for you guys. How low do you think, so let me give you three options. First option, do you think that a machine learning algorithm is basically nearly as good as a human, but not quite as good? Do you think, how many people think that's the outcome? You train these things and the machine learning option is going to be better than do just not as good, but close to compared to a human? How many people think that's the outcome? Nobody has an opinion yet. Here's the other two choices. Just so you're that's a good point. So what about the machine learning algorithm doing better than a human? Because that's another option. How about much worse? Those are your three. So how many people think nearly as good? Raise your hand. Okay, how many people think better? Okay, better. And how many people think much worse? Okay, hopefully, Zoom people, you can't see your votes, but maybe you have your opinions, so keep them in your head. So here's what we found. And the way we knew this was because we had a gold standard that we developed. We actually sat there and literally spent hours perfecting our scoring. So four of us basically quadruple scored every image and then went through it literally again and again and again. And when we did the machine learning classification, because the machine learning is better than humans, we looked back at the machine learning classifications and we re-evaluated whether we got it wrong. And in many cases, we got it wrong and the machine got it right. But our gold standard was basically, was it there? Do we know for a fact there was a flower there? So we had thousands of these images and we scored them ourselves to a gold standard. Compared it to basically a trained volunteer or a machine, that's what we found. So in some ways, really good, in some ways a little bit perturbing in terms of like computer and human intelligence, I guess. Right? Okay, so this last vignette that I'm going to cover here is really an extender from where I've been, in the sense that we spent all this time building this gigantic semantic graph store of phenology observations. And the first thing we started thinking about is like, that's a really amazing resource. And even before we built it, we were like, we have questions. There are questions about basically like, that can't be answered with remote sensing. You can't look at flowers with a satellite-based view of the world. We can't ask if flowers timing is getting earlier or later when in response to urbanization in your environment. You just can't do it, right? And you can't do it species by species. So you can't look at phylogenetic effects. You can't look at whether they, for example, if I wanted to know, does is sensitivity to change phylogenetically conserved or not? I can't answer that. I can't answer the question with the remote sensing data. If I want to ask if the timing itself is the time when you see the leaves on trees is phylogenetically conserved, I can't just ask that question, right? So that's what I wanted to know. We wanted to know from having these enormously powerful data resources to basically ask these questions. And so that's what we did. And what we really were interested in is basically how urbanization impacts phenology at the cross-continental scale. Previous work has definitely shown that we get earlier spring in urban areas and longer growing seasons. And everyone's argued this is related to the urban heat island effect. And in a neat way, urban heat islands like that is linear, right? Where you have more concrete and more heat absorption and more heat reflection. That's been seen as kind of an experiment in global change, right? Because I mean, basically the urban areas reflect what the world's going to look like in general down the road, right? So they've been used that way. It's kind of an experimental basis, right? But that work has been either by remote sensing data, which, by the way, if you look at the phenology remote sensing literature, there are real problems with some of the core like signal to noise ratios in remote sensing data. So having like actual observations on the ground to me is super important, right? So we were able to do this, so this has been done regionally, it's been done with remote sensing, but how do we put this all together in the big picture, right? That's what we did. We were able to gather effectively a lot of estimates of the timing for leaves to be unfolded on plants. That's just basically reflecting the spring green up signal, but this is like unfolding leaves. And for each of these pixels, these are individual estimates of the timing for leaves to appear on trees, right? And then we also did this with flowers, and like the flowers as well, like no one had ever been able to kind of gauge whether the flowering timing of urban plants is different in relationship to kind of the landscape and the climatic forces at this scale. So this is super simple, guys. So this is not rocket science at all. Like we had this data, and we had basically maps of human population density, which is a kind of a proxy for urbanization. You could argue there are lots of different proxies we could have used. We could have used a human footprint index. We could have used a impermeable surfaces layer. We chose urbanization proxies and human population density because we think that reflects where buildings are in the landscape in some fundamental way. Where humans are in terms of population density is where you're going to find buildings and other kinds of roads and resources, right? I mean, human footprint would be another great option here. And we had this temperature. We basically took temperature from November to May, averaged over a 10-year period, and had a temperature value. And then for every observation, we had a human population density value, and we had a temperature value. Super straightforward. So we basically were interested in asking, like, does regional temperature and urbanization basically interact to impact phenology across continents? And we also wanted to know if we could look at species traits and phylogenetic relationships here. Like, how does that matter for what we see in terms of the signal? And I'm not going to cover the trait side, but I will cover the phylogenetic side. And then, is there more than just urban heat out here? Is that the real story? Because people basically said it's all about urban heat. What about changing water flow in cities? What about pollution? What about all the other factors that are clearly happening that change how basically the core physiology of plants that live in urban areas? And we wanted to see if we found the same patterns for flowers and leaves. Okay, so what do we find? By far and away, the best model without question is a model. This model weighs of 1. In fact, these are by far and away the best models. Are interaction between temperature and human population density. What does that really? And you can see that these are the coefficient values for standardized linear mixed models, phylogenetically controlled linear mixed models in this case, using a package written by the core author Guralnick, who was also the lead author on this Nature Ecology Evolution paper covering this sort of urbanization and climate context results. What does it mean? Really cool. So urbanization advances spring in the coldest areas. So if you're living in Idaho, urbanization means that your springs are earlier. But if you're living in Miami, what it means is that spring leaf onset or spring flowers happen later, not earlier. And as you move into the subtropics and these warmer environments, basically these effects are definitively climate context dependent. And that pattern is actually really super strong and clear here. So again, like basically depending on the average temperature, the effect of higher population densities might be either positive or negative in terms of the relationships. Okay, so we also gathered land surface temperature, and we were able to basically look at deltas between pixels that were in the most urban areas and pixels that were in non-urban areas. And using those deltas, we could kind of measure the urban heat island effect and separate it from the effect of other potential factors that are happening in urban environments. So what was really interesting is when we use that delta as covariance models with human population density and temperature, we found that even with that urban heat island effect accounted for, we still found the same signal of population density being important in the models. And that implies is that it's not just the urban heat island that's driving these changes that are happening in human population dense areas. And that's really important because it says you can't just say that because there's a UHI effect, that's the key thing that is determining the urbanization impacts on plants. And it means that you can't just sort of use the substitution of cities for a warming globe, right? So that's cool. So we were also super interested in like the phylogenetic effects here, right? So like we have hundreds of species that we sampled for these models, or we're basically fitting linear mixed models with phylogenetic covariance structure, right? And so, but we can also look and actually test if you know the timing is phylogenetically conserved. But we can ask if the sensitivities are phylogenetically conserved. If sensitivities being phylogenetically conserved is super interesting. So when we talk about sensitivity, that you have a major response or a little response to climate forces. There's been lots of work, actually there's been not that much work, but there's been some work to suggest that's phylogenetically conserved. That's not what we found. So we find that the timing itself is conserved for sure. So mean flowering time and mean leaf out times are relatively highly conserved phylogenetically, but the responses, the sensitivities are not conserved phylogenetically, which is really interesting and important. And so knowing that that's the case, it means we can't just simply know a few species and assume phylogenetic conservation kind of rules here, which is kind of neat. All right, finally, I'm almost done, you guys. And I think I'm almost on schedule. I wanted to finish with a couple minutes to spare. We finally examined leaf senescence and growing season length. And this is a paper that's literally in that last revision stage where like the editor wants like, did you like dot your I's and cross your T's, right? So this is work where we extended this to think about growing season length and senescence. And so we had data on senescence, actually shown over here in green. We had data on season length where we were able to look at onset and offset and then measure the duration of the growing season as just the differential. And really, really interesting, the same pattern, no matter how you slice it for senescence and for growing season duration or length. But the general idea here is that if you're in a cold climate, the urbanization effect is going to mean that you have longer growing seasons. So if you're sitting here in Idaho, your growing season length is longer in an urban area than it will be in a rural or wildland area. But if you're sitting in Miami, it's the exact opposite. Here, you'll see that basically for high average annual temperatures, higher urbanization means that you have shorter growing seasons. Again, completely climate context dependent, how urbanization impacts plant growing season lengths, right? So super interesting and important. And what's kind of cool is we were able to actually look at the data from the in situ records and compare it to the remote sensing data directly. So here's the data for remote sensing across different land cover types, where you can see for cold areas of the eastern US shown in blue, this isn't supposed to be an indication of political affiliations. I recognize that using blue and red has like more complexity than I ever imagined these days. Maybe this is actually closer to the truth than I would care to want to suggest. But anyway, blue areas are cold. That's why it's blue, not for any other reason. Warm areas are red. And as you can see, like basically the same pattern where you see increasing growing season lengths over urbanization gradients shows up, and in warm areas you see that sort of weaker or converse pattern, right? So that all jives. Remote sensing data gives us one signal, then the CT gives us a different signal. They tell the same story. Cool. Okay, so what do we take home from all this? Well, urbanization effects are temperature context dependent, with implications really for how urbanization influences phenology across latitudes and across gradients in general. The urbanization effect is not just an urban heat island effect. And we really want to get that tropical data to sort of piece together more about what we can see about how urban environments impact plant phenology. It's a missing piece that we'd love to close with more data, right? More observations, more resources available. And we'd love to see more detailed local scale urban experiments. I think it'd be super fun to do. And there's a group of folks really jonesing to kind of get that done. And in general, where do we stop and how do we come cycling back? Time cycle is a loop that we close. How do we close the loop on time cycle for this talk? Well, I want to just say a couple things about where the meta story was and my journey was in this presentation. I really believe that we need to change science to be more team oriented, to be more collaborative and not competitive, where we need to work together to solve problems. We need to be interdisciplinary. I realize this is not the last version of the presentation that I put together, because on the last slide of the current presentation, this is one before it, there's a huge list of people, all the people, Daejeon Lee, Brian Stuckey, Laura Ben-Skelly, Stephen Mayer, Casey Youngflesh, all the people who led those papers. I'm not the lead author of those papers, but I think that, you know, intellectually the work reflects a team effort to bring together and to produce, and always the kind of person who wants to work on that kind of team. I want to be involved in making science happen that is collaborative and works around the idea of larger team solving problems and trying to push boundaries of what we can do, whether we're talking about data or ecological systems or temporality, right? So I'm a hybrid. I basically have two tracks in my career. One is very science oriented and learning how to kind of think about biology questions, and the other is more about working with sort of tools and building infrastructure. That hybridization has been great for me. It's made me, I think, something different than I expected to be in my career, but I'm really happy about that. And the real joy I take is the ability to bridge across communities and talk to ecologists, evolutionary biologists, stakeholders, all the people that kind of make up the work that we do in a way that will allow us to basically work collectively and collaboratively. And I really believe in this idea about continuing to grow the use of our community science and natural history questions data to answer questions about the planet. And with that, I am done. Thank you so much for your attention. This was so much fun. Thank you. What do you think, Kristen? Should I see?
Maybe check the chat if there are any questions.
Sure.
You see it too. There's like that.
Oh yeah, you got it.
Whoa. Oh, yeah. say so.
Lots of thank yous. Thanks to you guys for sticking it out to the end here. But I will, yeah. Go ahead.
I have a question. It kind of relates to the earlier part of your talk with respect to mismatch and phenologies. The obvious thing to me, and I'm sure you've thought about it, is there's different environmental cues that stimulate timing and cycles and things like that. And so, so what have you thought about that, and what have you, how have you kind of incorporated that to the mismatch studies?
Question. That's a great question. So we
You're trying to get to the bottom of beer.
yeah, yeah, I'm looking forward to beer already. So with the side of the story, one thing that I did not mention is that it turns out that the diapause strategy turns out to be incredibly important in how insects are skewing against the environmental signal that they get. So butterflies will overwinter as adults, pupae, larvae, or eggs. And we see a super strong decline in egg overwintering butterflies in the last 20 years. And it's strong enough where it's shockingly strong when you pull data out and you look at the patterns across those different diapause strategies. And how an egg overwinter experiences early season climate is completely different than how a larval people overwinter does. And it turns out larval people overwinters show dramatically different phenological sensitivities, and we're able to kind of get this sort of idea of like even under warmer springs, there's still sometimes delays in they're always earlier, but the amount of earliness varies, and you kind of catch into this sort of climate plant mismatch that then dramatically shows up in the curing related to the physiological responses of the eggs. So what a lot of my collaborators are doing is basically doing a lot of longitudinal experiments, getting the physiology of this. And I think to get there, you're going to have to build these large-scale observational data sets to give you a pattern that will tell you so much about all the processes. But getting those experiments built and out there and sort of get these data collected in a way, and then bringing some of that back into the lab, doing the experiments like you could do that were more tuned on the physiology side, the curing side. That's where we're going to learn more. I think that's the place where a lot of people are starting to really align. Let's get these broad scale flow experiments put together, because I don't think we're going to learn it in the lab. I don't think going in and just doing a physiological experiment allows you to tell you enough, or even the right signal. I think you have to be in the field collecting data across scale, and you have to let it up. But that's exactly where I think we need to go. And it's funny, we got this macrosystems funded work. We've been at it for 5 years, and what's really raised our board, like, oh man, we only have this next tranche of support to kind of tackle this thing. We could do so much. Like, one of the things we really want to believe in is that we could take adult phenology in abundance and use ecophysiological models to backcast caterpillar densities. Turns out it's super hard. But if we had the right data, and we had the right amount of species and the right amount of ability to use phylogenetic approaches to kind of fit the models better, we could basically make those predictions about how they have to do as much monitoring of caterpillars, which, by the way, is super. Good question.